So hello
Hello.
afternoon, good evening to us all. I hope we've had a wonderful day. It's been a pleasure to see you today. Thank you so much, Professor, for joining us today. Love to thank Dr. Midega, who is also among us, for her support in also getting to invite Professor Schofield for the session. So I think we'll just start and so that we don't take so much time, and then the rest will join us as we continue. So I'd like to hand over to Jimmy so that you can introduce our guest for the session. Then we can continue. Over to you, Jimmy, and welcome everyone.
Okay, thank you so much, Daniel. Thank you, everyone. Welcome to the session. We're so happy to have you at this moment. Thank you so much, Professor, for committing your time to attend this session. We're so much grateful for that. I also would like to recognize Dr. Janet Midega for being with us. She has been supporting us all through. Thank you so much for that. Welcome, everyone, all members in the house. I'd like to introduce our speaker today, Professor Christopher Schofield. Is the head of organic chemistry at the University of Oxford and a fellow at the Royal Society. Christopher is a professor of organic chemistry at the University of Oxford, Department of Chemistry, and a fellow of Hertford College. Professor Schofield studied functional, structural, and mechanistic understanding of enzymes that employ oxygen and 2-oxoglutarate as a core substrate. His work has opened up new possibilities in antibiotic research and oxygen sensing and gene regulation. Actually, we have an established researcher within us. Thank you so much, Professor, and feel welcome. I think, we can now start. Over to you, Professor.
So thank you very much for the kind invitation. Firstly, can you hear me okay?
Yes, we can hear you. We can hear you both.
Okay, that's great, Jimmy. So yeah, and thanks for the kind introduction as well. So I do remember just about what it was young and to be starting out in science. And I think that the most important thing I can do in my job is help to inspire and enable young people across the globe to have a career in science. And I do think it's a really fantastic time to be working at the interface between physical sciences and medicine at the moment. And I'm going to tell you a little bit about some of the things we're interested in our group. I am a chemist in a chemistry department. So one important thing to say is, I don't know the backgrounds of people who are listening today, but if you don't come from a chemistry or biochemistry background, and some things appear quite complicated, that's fine. That's the intelligent response. So don't worry if you don't understand everything that I say. Some of the things are moderately complicated. And I'm just going to share my screen now. So I'm going to talk about natural products. And I am a trained chemist, but I was always interested in biology and medicine for the usual family reasons. But I'm not very good at remembering long lists of things. So chemistry or maths was a little bit easier than doing basic biology or medicine. But I wanted to always apply the chemical knowledge that we acquired to biology and medicine. And it took me a long time till I felt confident to do that. So I hope we can reduce the activation energy of young people to do that. So don't worry if some things are complicated. Don't worry if you come from a chemistry background that the names in biology or medicine appear difficult. They were to me, and we chemists tend to think in terms of structures or mechanisms. So don't worry if you haven't perfected a language of the different disciplines yet. So I'm going to share my screen now. So yeah, Jimmy, can you see the screen okay?
Yeah, it's okay, Prof. It's okay.
Okay, all right. So I like a few things in life. One is music, and a famous rock and roll band with the Beatles. Maybe some younger people haven't heard of the Beatles, but they were, you know, arguably one of the greatest musical bands. My other enjoyment outside of science is football, and I'll get that in as well. But I also enjoy natural products, and for reasons I can't explain, I was really interested in mechanisms by which natural products are made. And so, yeah, as indicated earlier on, I think this is a great time to try and connect detailed molecular science with biology and disease. But that is complicated, stating the very obvious. We human beings and other animals are infinitely complicated in the ways that we behave and respond to things as well. So in my opinion, if anyone tells you that the underlying molecular science is simple or they have simple answers to things, don't trust them. You're young as well. So when people use jargon names like genomics or transcriptomics or things like that, don't trust them. Try and understand. You're young, you've got time on your side. And I'm going to make that point a number of times. So I came to Oxford to work on the penicillins on the left-hand side here. And these are natural products that have been honed by evolution, likely over a period of 1 billion or more years. One of my other favorite compounds, artemisinin here, which you're probably aware is the most important anti-malarial drug, is a fantastic natural product as well. And oxygen, the small molecule oxygen from the atmosphere, is crucially important in the mechanisms by which these molecules are made. I'd also make the point that there's huge investment over the last 50 years or so in what might be called modern medicinal chemistry. I do not think modern medicinal chemistry in the large pharmaceutical companies would actually discover these molecules now. In fact, the expertise that led to the discovery of the penicillins, which is what I'm going to focus my talk on, stems back to the mid-1940s during the Second World War, when there was a massive collaborative effort. And I don't think, in my opinion, the science is not present in the current large companies. So we do need new ways of doing things. So to emphasize, and this is also not a cutting edge talk, though I will tell you about some new methodologies as well. It's really a fan letter to natural products, which I enjoy, penicillins, and the pioneers in academia and industry. Some of the fantastic science that led to the drugs that we have today was done in industry, not in academia. Then the final point is really to people like myself who enjoy details. So if you enjoy the mechanistic details, I believe, and this is a belief, they really matter. And if you focus on something of basic science interest, in the long term, that will lead to something useful. So yeah, you'll be aware, and I saw the nice slide you had at the beginning here, it's been a long time since we had any breakthrough antibiotics. And arguably the last really breakthrough antibiotics was a natural product. This was a compound called daptomycin. And this originated from pioneering work done in Eli Lilly in the 1980s and 1990s. So it's a typo there on my slide. It was commercialized by another company. But around about 1990, Eli Lilly and the other major pharmaceutical companies really stopped serious work on antibacterials. Why was that? Well, the main reason was economic. So Lilly developed a compound called Prozac that was making $10 billion pounds a year. GlaxoSmithKline discovered Zantac, and Pfizer discovered Prozac. So making a new antibiotic that was only going to make them $1 billion or so a year wasn't of interest. Now, they anticipated they'd make these new wonder drugs, blockbuster drugs, every few years. That hasn't turned out to be the case. But this was fantastic science done up until round about 1990. So one of the things I think we want to get back to is the ethos when that collaborative science between academia and industry was done that led to the fantastic medicines we have today. But although I always wanted to apply my chemical skills to biology and medicine, I came to Oxford to work on the mechanisms because I was interested in mechanistic science. And this is me talking to you young people as sort of parent here. In terms of employment, you want to do something that you enjoy. That's really important because it's all very well wanting to save the world, but if you don't enjoy what you do on a day-to-day basis, you're not going to stick at it long term. Something that pays you enough money to live, and I'll make a point about that later on. And ideally, if it can be useful as well, that would be nice. So I came to Oxford to work on what I think is a fantastic reaction from a chemical perspective. It's catalyzed by an enzyme, and it's a reaction by which a simple dipeptide at the top here is converted into the penicillin nucleus. Now, if you haven't come from a chemical biochemical background, don't worry. This remains a fantastic reaction today, and there is no synthetic reagent that will do it. So this can only be done by natural chemistry. And then the main, not the only mechanism of resistance to the penicillins is addition of a water molecule to hydrolyze this bond here. And this four-membered ring is known as a beta-lactam. So you add water to it, and you go from the penicillin structure to the beta amino acid, which is inactive. And in essence, these two reactions, they look a little bit more complicated than that, but in this case, it's adding two oxygen atoms, and that might not be apparent, but that's what's going on. And in this case, adding a water molecule. So relatively simple reactions. So in the case of the reaction on the left-hand side, how penicillins are made, I'm not going to talk about that today, but from that basic curiosity, that took us into all kinds of different areas. How humans respond to changes in oxygen concentrations. If you go up a mountain, we make more red blood cells. Tumors are also hypoxic, and that's led to drugs. The energetics of oxygen, and that links to medicines developed on the basis of rocket fuel. Abuse by athletes, signaling in plants, ethylene. It's a small molecule that ripens tomatoes and bananas and all fruits. And even the science of so-called epigenetics. We all know that identical twins are not identical. And furthermore, it's my belief that human beings are not encoded for by sequences of 4 letters. That was a myth I was taught when I was your age. The central dogma of molecular biology: DNA goes to RNA, and then get proteins that are worker molecules, and DNA is a code. In my opinion, that is simply not true. So one message is, don't trust what old people like me tell you. So I'm going to tell you a little bit about the story of the penicillin natural products. Now, it's likely that the penicillins were used by human beings for hundreds, if not thousands of years. But in the modern age, Alexander Fleming famously made an observation that one microorganism, which happened to be a fungus, made something that killed bacteria, another microorganism there. And he published that observation just simply as an observation. And I think that's an example of where the publishing community works well. Today, I think publications have become somewhat of an end in themselves. And as a taxpayer citizen, I don't care where you publish, as long as you do useful science. And so that's a good example of how basic research can have unexpected future benefits. He just published that interesting observation. Then in the 1940s in Oxford, Florey and Chain and their co-workers picked up on this observation and started to culture fungi with a view to isolating the penicillin substance. And the structure of that substance was determined by Dorothy Hodgkin, who at that time was one of very few female chemists working in the UK. In my opinion, the greatest chemist to come out of Oxford. And diversifying the types of people that are doing science, I think, is something that we still need to work on today. And she determined the crystal structure of penicillin and demonstrated that it contained this 4-membered ring. Now, in fact, this 4-membered ring beta-lactam structure had been first proposed by Edward Abraham. And this is a picture of when he was old. When he did that, he was very young. He was in his 20s. And that was against the view of the bossy organic chemist who was in my job at the time, Robert Robinson. He's a very modest person. So he not only was the first, to my knowledge, to propose the beta-lactam structure, he was the first to determine the resistance mechanism to the beta-lactams, first molecular resistance mechanism by anyone. And he was a co-discoverer of these cephalosporin antibiotics here, which also contain a beta-lactam ring and are arguably the most important anti-infective medicines today. So the penicillins and successive generations of beta-lactam antibiotics, the cephalosporins, carbapenems, and monobactams, all contain this four-membered ring. And any way you count it, if you look at the blue bit here, they're more than 50% of antibacterials. So this is counting by lives saved, doses, commercial side as well. So one of the remarkable things about these antibiotics, and resistance is a huge problem, is how this particular chemical entity has survived to enable the most important medicines, probably along with steroids, small molecule medicine, I should say. Vaccines have always for long been the most important medicines. They've survived despite resistance. And in fact, as we'll see, there's quite a lot of rubbish talked about resistance as well. So how do they work? So why is that? Why is this particular class lasted for so long, whereas other classes have come and gone, sometimes not even making the clinic? So I think that's the kind of question that we can think about in academia. So one trivial answer to that is they were evolved by nature over a period of probably at least 1 billion years, and probably more than that. So nature has selected for particular compound classes. So I think that simple argument is a major case for natural products. So how do they work? So don't worry about the details here, as you're probably taught at school as I was. They interfere with a biosynthesis of the bacterial cell wall. And central to the formation of a stable bacterial cell wall is swapping of one amide or peptide for another one. You make a cross-link between the red bit and the blue bit here, and that's important there. The details don't matter. So in essence, this is another way of showing the same thing. You have one amide, and this reacts with the enzyme to give, in the chemical nomenclature, an ester intermediate. So this is an enzyme substrate complex. Then another amide comes in and swaps that to make the cross-linked species. So what do the beta-lactams do? Well, in essence, they join the two parts of the substrate together, so to disrupt release of half of the substrate at this stage. So the beta-lactams, such as the penicillins, react effectively reversibly with a nucleophilic, the reactive serine, at the active site of these enzymes to give a stable complex. Now, again, as I said, these are natural products that have been present in nature for a long time, and nature naturally evolved beta-lactamase enzymes that hydrolyzed the beta-lactam ring to give the inactive enzymes. And these are the nucleophilic serine beta-lactamases. And their trick was to learn to hydrolyze the same acyl enzyme intermediate that you have in the transpeptidase catalysis. So from a period actually stemming back to the 1950s to round about 1990, in industry, people searched for natural products that would inhibit the beta-lactamases. And this is a little bit counterintuitive. So these are the three key compounds that were found. Clavulanic acid, which is my favorite compound in the talk. It's never been made by synthesis in an asymmetric form. So just because something's small doesn't mean it's easy to make. And as an aside, for something to be useful as antibacterial, it has to be made for a price. So for a course for treatment of tuberculosis has to be a few $100s over a period of 6 months or so. So these compounds are counterintuitive because, like the antibacterials, these are the antibiotics here, the inhibitors also contain a beta-lactam ring. And that means that they can bind very tightly or efficiently to the beta-lactamases, but I'll tell you how they work as well. So actually work began during the Second World War, during the mid-1940s, to see if you could replace this beta-lactam ring. And that was achieved approximately a decade or so ago when this compound, avibactam, and don't worry, this ring, the red bit here, doesn't really look like a beta-lactam ring, but it works in the same way. So the key thing there is that it took approximately 70 years for the community to learn how to replace this beta-lactam ring. Now, why was that? Well, one, it wasn't trivial. And secondly, people didn't believe it would work. So, one of my other things is don't be afraid to have new ideas. They won't all work, but that's okay. So I'm going to tell you about that story and about our contribution to that. So secondly, if someone says something that you don't agree with, challenge it. So there's a famous statement by someone from the NIH in the late 1960s that the war against infectious diseases is over. So that never made sense to me because of the resistance mechanisms, and in most of parts of the world at that stage, people were well aware diseases like tuberculosis and malaria was still rampant. So this statement was clearly nonsense. So these are now some thoughts, because one thing I want to say as well is that although some of the results I'm going to show you have involved quite a lot of investment in terms of finance, the most important thing we can do is in academia is have new ideas. So some thoughts as to why the beta-lactams might be so special. And some of these might be trivial. I should say these are ideas, not facts. So firstly, they target something that's essential for bacterial survival. And that may limit resistance. And they react with an essential component of something that's essential. So also that limits resistance. That serine that I showed you, that's essential. So if you react that, you kill the activity of the enzyme. The penicillin binding proteins, or transpeptidases, sorry, another word for the same thing, are not present in humans. So if you optimize them, you were optimizing for a target that's not present in humans. That may limit side effects. Next, they're in the periplasmic space. So if you have microbiology, you know about that, that's fine. But bacteria have a membrane, and outside of that, they have a cell wall, and the space in between is called the periplasmic space. So the beta-lactams don't need to cross the cell membrane to be active. Now that's different for a disease like tuberculosis, which lives inside human cells. But for most gram-positive and gram-negative infections, you don't need to do that. Next, they react covalently with their targets. So they react to form a strong covalent bond. So why is that important? Well, I think one of the reasons is this covalent irreversible reaction means they're not reacting reversibly. If they're reacting reversibly, then resistance by efflux pumps could be more of a problem, because the efflux pumps can just bind the antibiotic and pump them out of the periplasmic space. So next, now this is a much more subjective argument. I'm an organic chemist, and it's very difficult to define beauty from an organic chemistry perspective, but I know what it's not. It's not a series of 4 or 5 rings stitched together by modern organopalladium chemistry. In terms of the penicillin there, every atom counts in terms of the structure here, in terms of binding to their targets as well. I'll make another point that as well. They're very densely functionalized small molecules. These are often more difficult to make than large molecules that are not just densely functionalized. I already made this point. This class of chemistry was validated by one microbe against other microbes very long before human use. There's over 500 beta-lactams being identified. And there will be more as well. We humans are changing the planet as well. Our use of antibiotics and the generation of resistance almost certainly means that natural product biosynthesis is evolving right now. This is a really important point. We used to think, or I used to think, that natural product chemistry was static. It's obviously not. Use of antibiotics and development of resistance means that natural product biosynthesis is emerging, which is one of the reasons why I think we need to make the arguments, the funding agents for more investment in natural products. Next point, it's not just the natural product itself that's useful. It's the combination of a natural product and the target. So I would argue that the beta-lactams are perfectly suited to inhibit their naturally evolved targets of transpeptidases. That doesn't mean they're suitable in all cases. So just because something is a natural product doesn't intrinsically mean that it's better from a medicinal perspective. So next point, they react with their targets as well. So yeah, ignore the detail on the slide here. So they're sufficiently reactive to react with the nucleophilic serine, and they react irreversibly. Now people used to make the point that the strained chemical nature of the beta-lactam ring was intrinsic to their activity. But that's not true, because these enzymes naturally react with amides. There's another example here as well of an amide that naturally reacts with an enzyme. So that's not so important, but they are chemically more reactive than normal amides. So actually, in terms of human use, I think that's quite important, because that intrinsic chemical reactivity means they have a limited half-life. So, you know, if you take a dose and you might take a dose of 1 gram or so of a beta-lactam, The chemical reactivity means that if you stop taking it after a week or so, there'll be no beta-lactam in your system. And that could be useful from a clinical side effects perspective. You don't want the drug to stay in your body for a long period of time. So I'll come back to this. So some beta-lactams as well, it's the same point as revealed by Dorothy Hodgkin's beautiful crystal structures. This is a chemical point here. This amide group here is sp2 hybridized for the chemist, but in the beta-lactam, this nitrogen is sp3 hybridized. That means this carbonyl group is more reactive. It's more like a ketone. So that reactivity is intrinsic to the activity. So you will have been taught at school, if not at school, at university, that catalysts work by lowering the transition state and hence the activation energy for a reaction. By the way, if you've ever thought these pictures are a little bit simple and it's a bit more complicated than the teacher is telling me, you're right. But we can discuss that afterwards if anyone's interested. Now, enzymes bind their substrates. They make a complex and lower the transition state. So if the transition state is lower in energy relatively than the normal reaction, if you make a mimic of the transition state, then that should bind tightly to the enzyme and mice inhibits as well. Enzymes tend not to bind their substrates or the products too tightly because of the inhibition there. So we'll come back to that point. So beta-lactams react covalently and near irreversibly to form stable acyl enzyme complexes. So evidence from infrared and other techniques indicates that when the beta-lactams react, you don't get a single complex. Now, why is that important? And this is beautifully shown by mass spectrometry. So if you take, and this I think was an early experiment that we did, if you take a beta-lactamase, and you can now analyze intact proteins by mass spectrometry and mix it with clavulanic acid, you get multiple different products. Now that's interesting because it means, well, you have that these products have different shapes. And that I propose means that it's more difficult to have resistance to something like clavulanic acid than if you have a compound that just fills a hole, which is much of without reacting, which is much of what modern medicinal chemistry does. And indeed, this is for the organic chemist, when clavulanic acid reacts with beta-lactamases, it almost explodes on the surface of the beta-lactamase. So initially you open up the ring and then you open up this second ring. And remember, this is not an antibiotic against most things, but it inhibits the beta-lactamases. You get a cascade of reactions that ultimately lead to an aldehyde. And the chemists on the call will know that aldehydes are more reactive than esters. So the hydrolytic water prefers to react with the aldehyde. And that's why clavulanic acid is a beta-lactamase inhibitor. One of the problems with a field like this is that's been worked on for a long time is that the funding agencies think everything is known. Don't worry about that. Yeah, another example would be the chemistry of DNA. Almost nothing is known about the chemistry of nucleic acids. Although there are tens of thousands of papers every year on it, exactly how water molecules and metal ions interact with DNA is simply not known. And there's huge gaps in our knowledge of how antibiotics work as well. Although people like me draw mechanisms were detailed in there, exactly how antibiotics work inside a person and the role of the immune system in antibacterial therapy is not known. But I just want to give you one example from our own work from recent years. Now, arguably, the most efficacious antibiotics are these carbapenems, although they have to be given intravenously. And these are good because they have a very broad spectrum activity, and they also show resistance to beta-lactams. And that resistance is because the hydrolysis of the ester is hindered because this hydroxyl group disrupts the water molecule that would normally hydrolyze this ester bond here. So we were studying the mechanisms of these enzymes. So next general lesson here is that when you're doing an experiment and it doesn't do what you expect, always follow that up. And usually it's because you've made a mistake or the company giving you the reagents has made a mistake, but always follow that up because it's the control experiments that don't behave very often where you learn new things. So this is a carbapenem here called ertapenem, and this is following the reaction by NMR. and chemists will know, you can look at the signals for these methyl groups here, which are doublets. And when you mix it with a beta-lactamase called NDM1, that I'll come back to later on, that's hydrolyzed, so you get very efficient hydrolysis. But when we mixed it with another beta-lactamase called OXA48, we got a new set of signals up here. And these signals, we weren't sure what they were, but the researcher who was doing the work in the lab did not ignore these signals. And ultimately, he showed that these signals originated from this 4-membered ring species here. So we've gone from one 4-membered ring in ertapenem to another 4-membered ring here. It's been rearranged and you get some hydrolysis. Now, why? So carbapenems have been worked on for, I don't know, at least over 40 years now. So this really should have been spotted earlier on and might have been. So it's led us to a new mechanism of resistance for this particular class of beta-lactamases. In the classical mechanism, you have hydrolysis, but in this new mechanism, the beta-lactamases, these class D beta-lactamases, rearrange the beta-lactam to give the beta-lactone, which is not nearly as active. So that's an example of something, yeah, don't be afraid to go back to old, but really important fields. So next thing, can you replace the beta-lactam, beta-lactam ring with something else? And again, this work goes back to the 1940s. So work was done, and I did some of this work with my own hands. So another important lesson here is sometimes it's difficult to escape the work that you did for your PhD. So I made in Jack Baldwin's group compounds in which the 4-membered beta-lactam ring was replaced with a 5-membered ring. And that compound was given to the microbiologist. I couldn't do the microbiology then. And I'd spent 2 years making the compound, which felt like an impossibly long period of time back then. And the results came back the next day. Sorry, your compound is not active. So this was disappointing to me. So I resolved to find out, why it wasn't active. And we did that over a period of years. And ultimately something useful came out of it. Now we did eventually in Jack's group make antibiotics that work like beta-lactams that didn't contain a beta-lactam ring. And we did that by introducing double bonds into the ring structure. And our rationale for that was we were making the lactam more reactive here by putting ring strength in. But as I told you, that's not necessary because the natural substrates are AMRs. So following on from that, and this is a chemistry point again, I started to think, why beta-lactams? I was into this question of why beta-lactams might be special. And from a chemical perspective, it's easier to make a 5-membered ring than a 4-membered ring. So the idea was that nature had evolved to use 4-membered rings, which are more difficult to make than 5-membered rings, in part because the equilibrium between the 4-membered ring and the ring-opened inhibited form was more over to the right-hand side for the 4-membered ring than for the 5-membered ring. So we tested that hypothesis. If that hypothesis is true, sorry, slides jump. If that hypothesis is true, if we do something to this species to drag the equilibrium over to the right-hand side, we should be able to make an inhibitor. And indeed, with time, we did that. So we made 5-membered rings analogs of 4-membered ring where we put electron withdrawing groups on the nitrogen here to drag the equilibrium over to the right-hand side. And you can fuse the 5-membered ring to another 5-membered ring, and that drags it over to make super potent inhibitors of nucleophilic enzymes. So we did that work in the early 1990s, and the truth is nobody was interested in it. It was not published in a fancy high-impact journal. We even saw crystal structures with a model nucleophilic enzyme, a protease called elastase. With this is the inhibitor here in gold, here overlaid with the crystal structure of the substrate there. And nobody was interested in the work, I thought, but actually that wasn't the case. That paper, although it didn't have many citations, actually as much as I cited some other work we've done, did turn out to be useful. Now, if what I told you is true, you would have thought that nature might have made non-beta-lactam analogs working like beta-lactams as well. And indeed, nature has done it, but to date so far, to our knowledge, only once. And there's another fantastic natural product called lactivicin here that was isolated by Japanese workers in the 1980s. We demonstrated the mode of action of lactivicin. And in the case of lactivicin, this extra lactone ring here drags the equilibrium over to the right-hand side, exactly in a way or in a related way to which our model compounds work. And again, we determine crystal structures to validate this mechanism of action. So that was academic, and we come back to these natural products. So in industry, this work had not been completely ignored, as it turned out. And in industry, workers in France, and ultimately this project transferred into AstraZeneca, where the production scientists and AstraZeneca did a fantastic job to make this compound for a price, develop this compound, which is now called avibactam. So this works via the, and we know that they read our papers as well in terms of mechanism. They designed this compound, and this works via reversible reaction, exactly as our mechanisms, but the equilibrium is dragged over to the right-hand side because the strain in this bicyclic system and the electron withdrawing group here. And again, we've worked the mechanism of that. So that work demonstrates that in new classes of natural product-based inhibitors, all of this work is natural product-based, can be done, but it does take time. Now moving on, there's a second class of beta-lactamases. I've told you about the nucleophilic serine beta-lactamases, but there's another class that instead of using a nucleophilic serine, uses a zinc at the active site. And this class of enzymes came from a completely different origin, and they have a different crystal structure. And for a long time, I thought very different mechanism, but now I've changed my mind on this. So these zinc enzymes use a zinc to polarize a water molecule to give a zinc intermediate rather than a covalent linked one. But is it really, these classifications of covalent and non-covalent are somewhat arbitrary to polarize the water molecule. Now, whoops. Now, I just want to tell you now, this is, I do want to emphasize the point that the most important thing in research is new ideas, but sometimes governments invest money in expensive toys that we like to use. And when we have been used, this is the cutting edge in terms of our research, is using a technique called X-ray free electron lasers. And this is the first one that was built in California. This is a kilometer-long laser that gives very powerful laser beams that you can use to collect diffraction data on very small crystals and collect diffraction patterns on a femtosecond timescale. That's faster than biology works. So we can use these to get time-resolved mechanistic studies on enzymes. And we've used this to, this is our current basic science, to look at the reaction catalyzed by which the dipeptide goes to the penicillin structure. So I just want to say this is now an example of very basic science in our group that I don't think will be useful in the short term, but I think it will be useful in the long term. And I just want to show you one picture from that. So this is an experiment where we take the enzyme substrate complex anaerobically, then we expose that to oxygen and see what happens. And then we can trap the product in the enzyme there. So this looks a very complicated picture. So please don't worry about the details here. Red bits are hot, blue bits are cold. So just focus on this helix here. If we expose the complex to oxygen, you can see bits of the structure get hot. And then at the end of the catalytic cycle, this helix is coming down, it's getting cold again. So this is really interesting because it means that the catalysis is a property of the whole structure. It's not just individual residues and the active site. And now why is this important? It's important in terms of resistance because many of the mutations we see are away from the active site. Probably many of us use biological washing powders as well. Most of the mutations optimize those are away from the active site. So this is sort of very basic mechanistic science and I hope one day will be useful. So there was a need to make inhibitors to these new metallo-beta-lactamases. Now the problem there at the moment is there is no money in this for the pharmaceutical companies. So I think this is something we can do in academia. And one thing we were interested in that I didn't think would be possible would be, can we make inhibitors that inhibit both the nucleophilic serine and the metalloenzymes? And I told you they're very different mechanisms, but our strategy was to ignore the protein and just focus on the mechanism. And in both cases, that goes via a tetrahedral intermediate. So if we make analogs of this intermediate, We just might have compounds that inhibit both the nucleophilic serine and the metalloenzymes. And what we did, the story I'm going to tell you about is boron chemistry here. But again, boron, how long has boron been used as an anti-infective? It actually goes back to the middle of the 19th century. So now, complicated slide again. Don't worry about any of the details except compare the two pictures here. So this is a cephalosporin, the tetrahedral intermediate for the nucleophilic serine enzymes and the nucleophilic zinc enzymes. With hindsight, could all of this work been done without any of the clever crystal structures? Yes, it was the idea, but just do chemistry. So the classical, again, please don't worry about the details here. The class of compounds we've come up and we're most excited about are these bicyclic boron compounds here. It took years to develop the synthesis to these compounds, which is non-trivial. But ignore the numbers. The most interesting thing here is they do inhibit both classes of enzymes. They inhibit the nucleophilic serine enzymes, forming a tetrahedral complex there. So the nucleophilic serine reacts with the boron. They react with the penicillin binding proteins. They're not fantastic antibiotics yet, but we're working on that. But they're very potent beta-lactamase inhibitors. And perhaps most pleasingly, at least a little bit surprisingly to me at least, they inhibit the metallo-beta-lactamases, again by forming a tetrahedral complex, but this time in a manner that bridges the two zinc ions. And I should say again, if you're a trained chemist like me and somebody flashes up these structures and you think this is really complicated, don't worry, that is the intelligent response. We've been trained to think in terms of functional groups, and there's just too much functional group chemistry here to think about. So the important thing is that the boronates mimic this tetrahedral complex there. So one boron-based inhibitor of beta-lactamases has been approved. That's Beta-lactam. It's not a great compound. It's a monocyclic compound. We think the bicyclic compounds will be much better compounds. So just a few conclusions from the sort of science part of the work. Then I come back a little bit to the career advice politics. So potent, bifunctional inhibitors of serine and metallo-beta-lactamases are possible. It just took more than 70 years to do that. Could it have been shorter than that if people have been brave enough? Yes. The academic work we've done, we haven't developed in terms of drugs. We never made the final drugs, but we've laid the groundwork. I hope for other people to do that and pioneered mechanisms. And that's led to tools. This is a challenge for the young people. How many other functional groups with the efficacy of beta-lactams are out there? My prejudice is to go for natural products, but people might design things chemically. I think the biochemistry of resistance is a fantastic field to work in. So it's not just antibiotics, antimicrobials, antifungals, antivirals, and of course, COVID is a huge thing as well. And by the way, I didn't make a slide on this. Beta-lactams can be used to treat COVID as well. Anti-cancer drugs and resistance in herbicide and farming is important. So I'll come back to that. I'll come back to this as well. Combating antimicrobial resistance with breakthrough drugs requires credible investment in both discovery and process synthetic chemistry. And as young people, I think you should focus on basic science. Ask for the money that you need to do the job. One of the problems is that academics tend to exaggerate what they do to get the money for career reasons. I want to emphasize that this work is highly collaborative. We've worked across Europe and LinkedIn with researchers in Africa as well, in South Africa and elsewhere. Jürgen here is from Romania, Martin from Lebanon, Andy from China, Anne McKenna is a Rhodes Scholar, came from Kenya as well. So this work has to be global if it's going to succeed as well. And some of the chemistry industry was also very supportive as well. So for me, in terms of career, focusing on detail is fine as well, but collaboration is also good. So a few sort of general career points to finish up with. You're young. Time is more important than having lots of funding for you at this stage. The most important thing is new ideas and approaches. Be brave. Don't be afraid to call out older people like me when you see things that you don't believe in. In addition to my talk has been science, and I'm a lab person, but I think it's really important to think about science and medicine politics as well. There is no need for TB to happen on this planet. This is a political issue, and we can work to rid TB from the planet. In terms of antibiotics, just to say a few things about that, more than 75% of antibacterial use is in farming. So it's not just human, but this is a key cause of resistance. And more than 75% of the antibiotics used in farming are human antibiotics. Now, we can't stop that. I'm not saying that should stop quickly, but the politics of food and the science of food is a fantastic field to work in. So going forward, and there's remarkably little research gone on food science. We've recently had a donation from a company called INEOS, and a key focus of that work will be, which will be internationally in scope, will be to look at use of antibacterials in farming. So finally, just to finish up where I started, so I do think natural products is a fantastic field to work in. Will there be compounds out there that have the importance to human science as the penicillins and artemisinin? I don't know, but I think there's a good chance there will be, and you should look for them. And yeah, one of my heroes in science is somebody called von Neumann, and it's a famous quote from him: "The scientists don't try to explain, they don't interpret, they mainly make models. And a model is a construct with the addition of certain verbal interpretations described of observed phenomena." So this was saying human beings are not encoded for by sequences of letters. There is much more to resistance than simply enzymes as well. But I do think it's a fantastic time to try and understand biology from a chemical perspective. It's an interesting field to work in. And occasionally, if you stick at it, it's important to stick at it for a long time, useful things will come out of that field. Yeah, I like football as well. This is my, I'm a Liverpool supporter, my favorite footballer as well. So, but the real cost of science to society will go down. You know, the really top footballers are projected to earn several £billion during the course of their careers. And that's justifiable or not, it doesn't matter. The point is that the science, the underpinning science to cure, to rid the world of things like TB, the money is there in society. It's a political issue as well. So it will be an increasingly good time to be in the field. Thank you for listening so patiently. And I'm happy to take any questions that you might have, either on the detailed science or more general things.
Okay, thank you. Thank you so much, Professor. Thank you so much for the fantastic presentation I've given. So, as young people, we are so much encouraged. As much as we are challenged how to come up with these inventions, we are encouraged into the field. Thank you so much for that. Now it is the time for questions and answers session. So I'd like to ask anyone with a question, to let out their questions. Thank you.
Hello, can you hear me?
Yeah, I can hear you fine.
Yeah.
I have one question for Professor Chris Schofield. I've always wondered in the era of drug resistance whether there is a single compound, particularly a natural product, that could disrupt the problem that drug resistance or antibiotic resistance presents today. Is there one structure that can work across every group of bacteria? Thank you.
Yeah, that's a really good question. So I think the answer to that is that there is unlikely to be one approach, and I do use the word approach rather than structure. So there is, so firstly, there's rather confusing nomenclature here between antibiotic, antibacterial, antiseptic, and disinfectant. So I think there is unlikely to be one molecule that will be a wonder drug for humans. Now, if you take bleach, for example, that will kill all microbes, but contrary to what the head of the ex-head of the United States said at one stage, that is not a good approach to take orally for humans. However, will there be new classes of drugs to be discovered that have the very broad efficacy and safety that penicillins have? I think that's likely the case. I think it would be arrogant to think that we've discovered all the fantastic medicines from natural sources out there at this stage. But I do think it's not just about drugs as well. And you know, as many people be aware that the most important thing we can do in terms of infection is raise living standards and provide people with clean and healthy environments. So the answer to your question, it's a really good question, Janet. I doubt there will be one compound, but will there be world-changing compounds to be discovered along the lines of the penicillins and artemisinin? Absolutely, that has to be the case. But it needs young people to go and find them.
Thank you very much.
Thank you. Okay, thank you so much, Prof. Maybe I'd like to ask one question. How do you identify the protein, the structure of a protein, or any natural molecule? How do you identify the chemistry of that molecule?
Yeah, that's a really good question. I had a slide I didn't show, which was challenges in natural product discovery. So isolation of natural products is an issue, and again, there's a question. So most really good antibiotics, firstly, we don't know the details of how they work inside cells. Most of them have multiple targets. We also know that in many clinical situations, more than one drug is often used. So could there be, now the problem with controlling mixtures of natural compounds, which people go back to, is just controlling them, quality controlling them, etc., is difficult to do. So how are natural products isolated? So the current key techniques are, well, comparison with standards is still important. So having standards, libraries of standards is important. And then techniques such as mass spectrometry, NMR, and crystallography are used. Now, a risk in the natural product field is that one will discover a compound that's already been isolated. And that was one of the reasons the company moved away from it. So in my opinion, well, what would I like to see across the world? In terms of, I'm a natural product chemist, what would my dream to be? I would like to see a series of linked international centers focusing on natural product chemistry. I believe that research on drugs and medicines, where possible, should be done in environments where those medicines are most important as well. So in Africa, I would like to see one of these centers in Africa, to be blunt. I don't know where in Africa. You can discuss that. Where there are collections of compounds and the expertise to analyze those compounds by techniques such as mass spectrometry and NMR.
Okay, thank you so much, Professor. Another question, as far as antimicrobial resistance is concerned, we have seen that these targets that are usually targeted by these antibiotics or the antimicrobial agents, the microorganisms are mutating the targets. Now, how can we modify our structures, our molecules, our drugs to conform into the structure of the modified targets? Thank you.
Yeah, that's an excellent question as well. So resistance comes by different mechanisms. One of that, as you say, is by target modification. Another is by enzymes that modify the drugs and efflux pumps, etc. So I think it is possible to start to use the structural biology that's available and to help to design existing drugs so they work to address the question of target modification. And again, this is relatively designing the molecules is a relatively cheap thing to do, because the crystallographic information is available from the protein data bank. Modeling techniques such as AlphaFold, and there will be, you may have heard of, that predicts protein structures, and there will be better versions of AlphaFold coming along, means that this information is available to everyone, and computational programs can be used. So doing computer chemistry, I think, is a good thing to do. I'm an experimentalist at heart, so I think there's a limit to how far you can go with computer chemistry. But I think that's something that's relatively inexpensive, and I think young people should do computer chemistry. Jimmy, but to make a more general comment, although we've these fantastic techniques have come on, the problem at the moment is, in terms of resistance, that many organisms have so-called multiple drug resistance. They don't just have one resistance gene, as was the case in the 1970s. They have multiple resistance genes, and moreover, nature has evolved machinery both in cells and outside cells for swapping these resistance genes. So I think this is a challenge. So in my opinion, there needs to be a combination of new things: discovery of new drugs, including natural products, and also synthesis as well. There has to be control of antibiotic use as well, because there is good evidence that if you stop use of an antibiotic in a particular region, resistance to that does start to go down. So further work needs to be done on that. And a good example of that is colistin resistance. My colleague at Oxford, Tim Walsh, together with collaborators in China, persuaded the Chinese government to stop using colistin, which is a last resort human antibiotic in animal use, at least in China. Now that colistin use has gone down, resistance has gone down. So you might say that's an obvious thing, and maybe it is, but it's a very simple and important lesson. So I think we both need to look after use of existing antibiotics and develop new ones. And may or may not be aware to people on the call that this is not scaremongering. There are actually very few classes of antibiotics, probably less than 5 or 6 really important classes in total. So time is running out. I did make the point about antibacterial use in farming. And I'd just like to say, this is absolutely not the use of the farmers. I think there's quite a lot of hypocrisy from some countries in the West here. So in the UK, for example, antibiotic use as a growth promoters in farming is banned. However, if the animals are sick, you can use the antibiotics. If you have intensive farming, animals will always be sick. The fundamental problem of antibacterial use in farming is the nature of intensive farming. Why is there intensive farming? Because the profit margins on food are very small. So I do think the case of use of antibacterial use in food is something where there's political, economic considerations need to be part of the dialogue, the conversation as well. And from my perspective, we have to accept that once society can afford it, the price of food should be increased to decrease the use of drugs in food production. But that's an opinion. Sorry, I gave you a long answer to that question, Jimmy.
Thank you so much, Professor, for the wide elaboration of the answer. Thank you so much. We have another question from Dennis. What is the bottom-up approach to chemistry? Over to you, Prof.
All right, that's a good question. Is that from Denise, is it? Okay, that's a really good question. So don't be, it might seem in some ways that I've done a lot of stuff and been a successful person, but I started off at the bottom. I was interested in reaction mechanisms. So the bottom up, if you're just interested in chemistry because you like it, I think you should do it. And that's really understanding reactions, in my opinion. There's other aspects to it as well, but understanding reaction mechanisms and contrary, one of the problems for your generation is there just appear to be so many facts out there. There is so much stuff to learn. And in my opinion, one of the tests of when you know a field is when you can say what isn't known. And there's probably less known about the details, the real detail of chemistry, than people might think from what your teachers and lecturers and textbooks tell you. The precise nature of what a bond is, for example, is not so clear. How things diffuse through solvents in complex environments, such as biological environments, I know there is very little known. And again, I don't know, I can't remember how long ago it was now, about 25 years or so, I said, we're working on these enzymes, and I don't think it's really known how the substrates diffuse through biological solutions to get to the enzymes, because they're bumping into lots of different things at the time and making transient interactions. And he basically said to me, oh, that's not a problem at all. It made me feel like I was stupid, was the truth, for not understanding it. But in fact, I know I wasn't stupid. Very little is known about diffusion in complex environments. That was just an example. If you don't understand something and you want to really understand your reaction mechanisms, I think you should do that. You might have to stick at it for a long time before you do anything useful from the drug perspective, but just wait and be patient. If you're studying, how even how water diffuses through water is an interesting question. So if you're interested in that kind of thing, I'd encourage you to stick at it.
Oh, that's wonderful, Professor. I'm so happy for the wonderful presentation you've given us all, and the question, for answering the question. So,
Hello.
Hello. Hey, Dan. Hello.
Yeah, I have a question before we sum up. Yeah.
Okay.
So sorry, I wasn't seeing the cursor of raising the hand. Yeah. So thank you so much, Prof. Chris, for the wonderful presentation. I've really learned a lot. As you're presenting, I realized there's so much that is happening, and most of us even don't know. And I'm really glad that you are recording the session. At least you'll be able to share it. My question is more generalized. Currently, the information out there is that, we are at a dead end in terms of drug research and development. But from what you've told us, there's so much progress that is ongoing. There's so much research that even if we can't see the immediate outcomes at the moment, at least there are some things that are building up. How can we, disseminate this information? I acknowledge maybe some of it, there might be some intellectual property issues, but the one that is free access, especially maybe research done in universities, how can we try to disseminate it across so that maybe we have someone who is interested in pursuing drug research, but because from what they read, from what is mainly available, they just get the idea that there's nothing that you can do at that moment. So how can we disseminate this information? Thank you.
Okay, so Daniel, that's a really, really, really good question. So firstly, please don't be intimidated by my talk, right? It might give you the impression there's a lot going on, but there really isn't in this field compared to the importance of the field. So I came back to this field. I went off into that other area I showed you one slide on, because for career reasons, that was good for me. But I came back to this field 12 or 15 years ago, seriously, because I believe it's more important than some of the other clever stuff. Now, in terms of the beta-lactams, just to give you one example, in terms of the chemistry, I'd say there was 3 groups in the world at that time doing beta-lactam chemistry, and it hasn't changed much. I'd say there's 4 now, it's us. And that was 2 groups in the US, both of whom the investigators were over 70, and Shionogi in Japan. There's a few other people, but seriously doing it. So do not be intimidated. You are the future. You've got the time, right? So yeah, if you're passionate about a field, go into it, and there's a huge amount to do. yeah, my advice is, it's a question about bottom up. It's a good one, because for research wise, my advice is either start with bottom up, that's fine, or start with biology or society, identify a problem, which could be AMR or global warming, whatever. Go to the extremes and then work back from that. Intellectual property, I just completely ignore it. This is my personal advice. Unless you want to be a lawyer, that's fine, and we need lawyers as well to think about it. But I think people talk about patents and stuff. Just don't worry about it. Just do. This is me speaking as a citizen, if you like. I just want you to do good science. And patents are there to serve society. They're not. It's a myth. They're not there for the companies to make money. They were a legal, I don't know what the right term is, construct that was invented such that groups of us would do useful stuff for the rest of us. And if people are incentivized by money, that's fine. I think one of the problems with the patent system for some diseases is that the time scale is just not long enough. And this is a problem with the companies as well. They don't work on a long enough time scale. So back in the day, and I'm talking like a very old person now, I know, when saying things were good, the companies worked on a time scale of decades. Now they work on a time scale, at least the publicly owned companies work on a time scale of the chief executive officer, which is about 5 years. And that's just not realistic. So if you want to make a new medicine, totally new class of antibacterials, we don't know how long it will take. If you want to make a medicine for stroke disease or mental disease, and drugs are not the main solution for mental disease, I should say. There is no companies working on mental disease now, because the length of time it takes is more than 20 years as well. So as an aside, I think the future of the drug industry is not going to be just pills and injections. It has to be linked in with environmental therapy as well, particularly for things like mental disorders. So if you want to think about, and this is important as well, the patent system is not working to deliver the medicines we want, or society. It's not the fault of the companies, just like it's not the fault of the farmers that there is antibiotics so used in farming. These are political issues. So thinking about ways that we can incentivize people to behave well for society and to do useful things long term, I think is a good thing to do. And that's a research project as well, trying to think about how you'd reinvent the patent system or get rid of it is a good thing to do as well. Finally, I used to be in awe, and still am to some extent, of the quality of the science that went on in industry. And there are still good companies doing that, but the number of people doing basic research in these large companies has gone down. So I do think there is a case for public investment in, for example, antibacterial drug or treatment. I prefer treatment to drug because it's not just about pills as well. And setting up publicly funded institutions. And sorry, this is a general point as well. It's important to remember that everything is publicly funded. This divide between public and private is a completely artificial one. We're a community. The taxpayer funds the roads that the companies use. We, fund the young people that go into pharmaceutical companies, etc. So this public-private thing is an artificial divide. We just need to have, I don't know, laws, or whatever the right term is, regulations that help the research, that make sure the research that we want as a global community happens. Sorry, that was a long speech there in answer to a good question.
Thank you. No, it's really important. At least I've really learned a lot. Yeah, and I think something that I'll try to see if we can also reinvent the patent system.
Yeah, please think about that. That's really important. I mean, it doesn't work at the moment. It's a myth that you need a patent to make money as well. Most antibacterials are off patent now, generic companies doing it. If a new penicillin was discovered and that was just published in the academic literature, or a new artemisinin, right? That would still be commercialized if it was good enough. When people complain about the you're having to have a patent. That's not true. That's just not true. If the medicine is good enough, it will be commercialized. Or translated, I should say. Maybe I shouldn't use the word commercialized because it doesn't have to make money. It could be not for profit.
Thank you so much. Over to you, Jimmy.
Thank you so much, Daniel. Thank you, Professor. Thank you, Professor, so much. We have done a lot, actually. As young people, we are encouraged. We're being inspired. Now, I think I should take this time maybe to invite anyone with any other question, before we close up the session.
So Jimmy, just finally, thank you again. It's been a pleasure speaking to you all. Really good questions as well. And I hope, some of you will stay in science and some will go into politics, economics, whatever you want to do. And just be brave and keep going and you'll be fine in the long term.
Thank you so much, Professor. Thank you so much a lot. As young people, we have learned a lot. Actually, you have taught us a lot of chemistry. It is good that to understand anything that surrounds us in an environment, it is a good thing that we learn the chemistry of it. For us to understand the drug that you are using, we have to understand the mechanism of them. The mechanism of action of any drug, we have to understand the chemistry of it. For the diseases, you want to understand the chemistry of all. Actually, it is so important. It's a very nice, interesting discussion today, Professor. Before I proceed, I'd like to invite Dr. Janet Midega to say a word for us. Over to you, Dr.
Hello, Jimmy. I think maybe she hasn't heard you. Dr. Janet, are you there? We'd love you to say something as we close off. And
I'm not sure Janet can join, Jimmy.
I think maybe she's having some internet issues. Because she has just left off the call. So thank you so much, Professor Chris. We are really happy. I know we have extended a bit past time. It has been a wonderful conversation, and really would love to have you once more, and so that at least we can know some of the wonderful work that you're doing there at Oxford, and also share more. I know there's also more that will now start happening, especially after I saw about the Oxford partnership with INEOS. So thank you so much, and so I hope that we'll have some people also to partake the route too. So thanks so much, and have a wonderful evening. Over to you, Jim.
Thank you so much, Prof. Thank you, Daniel. Prof, there's something I would like to mention before you close the session. This is a group of inspired researchers, a group of students in Africa. Actually, mainly we're working against antimicrobial resistance, and as you also struggle in our research, maybe we are open to any other opportunity that is up there in Oxford and any other place. So maybe you just let us know in time. And we will be happy to have you back in time any other day, any other time. Feel welcome again, Professor. I don't know if Dr. Midega is able to say something.
So Jimmy, I think she's left the call, but thank you. I mean, so firstly, I appreciate that you guys are working on a Saturday evening, I think, for you as well, which is good. It means you're committed. It would be nice afterwards. I'm sure we can do something together, Jimmy. And if you email Janet, or you and Daniel email Janet afterwards, we'll try and make something happen. Okay, I don't know what it will be, but we'll do something.
Yeah, true, Professor, sure.
All right, so I don't know if you know, but I met Janet earlier on this week. She works at the Wellcome Trust in London, and I had a very good conversation with her. I can't remember what day it was now. Wednesday this week. Yeah, we can have a chat with Janice again, and we'll make something happen. Okay.
Yeah, Prof. True.
Thank you so much.
All right. Take care, everyone. And yeah, have a great evening. And yeah, good luck in your careers as well. You're going to do great things. I know that.
Yeah, thank you so much, Dr. Chris, Professor Chris, and bye-bye.
All right, bye-bye.
Bye, Prof, and have a nice time.
Yeah,
Welcome
and
again.
you take care. It's been a pleasure. I've really enjoyed it.
Thank you so much.